Namaste students. I am JVN Dr. Aziz Mohammed Khan from Department of Food and Biotech. Today I am going to discuss about the animal cell culture. That is the introduction of animal cell culture. What is the meaning of animal cell culture? So in the biotechnology process, there is a technique that is known as the plant tissue culture, animal cell culture, animal tissue culture. So these are the terminologies, and according to them, we need to understand what is the culture. This is the technique in which we grew any cell, okay, any cell in the artificial medium that is known as the cell culture technology. So if we are growing any cell into a artificial medium, then it is known as the cell culture technology. If we are growing any tissue, then it is tissue culture technology, and if we are growing any organ, then it is organ culture technology. Depending on the type of cell, we can name the type of the technique. For example, if we are going to culture animal cell, then it is animal cell culture technology. If we are growing any plant cell in the in vitro condition, then it is plant tissue culture technology or plant cell culture technology. So cell culture is the process which refers to the cells grown in a controlled artificial environment. It means they are grown in the in vitro conditions. Okay. So it is clear that what is the animal cell culture? Animal cell culture is technology which is used for the growing of animal cells in the artificial medium in the in vitro conditions. Okay. So on the basis of these animal cell culture technology, there are two types of cell culture technologies. Okay, the primary cell culture and the secondary cell culture. But first of all, we need to understand what is the requirement of any cell culture technology. For any cell culture technology, we need some artificial media. Okay, any cell needs some specific media. And if we are talking about the animal cell culture technology needs more complex media than any other uh, media. For example, if we are talking about the microorganism, then microorganism and do not need any complex media. Simple media can be used. And if we are talking about the plants, and then plants can be also grown very well on the in vitro conditions, but the media is more complex than the microbiological media. But when we are on the way for the animal cell culture technology, then the media becomes more complex along with the environmental conditions, because this is the animal cell, this is the animal tissue, which are very hard to grow in the artificial medium. So these parameters must be maintained. The another thing is that the environmental conditions is also another requirement which need to be conducted, which need to be monitored. Okay, so on the basis of these technology and the uh, culture, there are two types of cell culture. The first one is primary cell culture. For the primary cell culture, first we need to isolate a cell from the tissue. Okay, first we need to isolate a cell from the tissue. When we have isolated a cell from the tissue, then this cell need to be sterilized and it need to be cultured in the synthetic media or media, natural media. So after isolating, uh, the first when we culture and inoculate these cells in the media first time, okay, first time, then it is known as the primary cell culture. Okay, the cells which are dissociated from the parental tissue, and when they are grown in a suitable medium are known as the primary culture. Means the first cells which are grown in the artificial media are known as the primary cell culture, and these cultures are uh, com comprised of different types of cells, and they are dividing cells. Okay, they are dividing cells, and they will dividing only for a limited time. They will not divide for a longer time. Okay, so on the basis of these cells, there are two types of on the primary cell culture, there are two types of techniques can be utilized. In the first technique, adherent cells are used. What is the meaning of the adherent cells? These are the cells which are attached on the any surface, that the surface might be solid or a semi-solid surface. Okay, so the cells are attached on a solid media or a semi-solid media. Okay, so the need is support for their growth. These adherent cells need a support for the growth, and they are attached on a culture vessel with the use of some extracellular matrix. Okay, and this immobilization is known as the immobilization of these cells, and these cells are adherent cells in the culture media. Okay, the example are the fibroblasts and the epithelial cells. The next type of cell is suspension cells. When the cells are grown in the media in the floating conditions, okay, they are not immobilized on any surface. When they are in the floating conditions, then they are known as the suspension cells. Okay, they are grown in the floating in the culture medium. And the example is the hematopoietic stem cells, such as the stem cells derived from the blood, spleen, and bone marrow. So these are the examples of these suspension cells which are in the floating conditions in the media. Next is the when we need to culture these cells. So as we know that uh, these primary cell culture can grow for a limited a time, and they can divide for a limited period of time. So when that time reaches, when there is no growth is observed in the cell, then these cells will be subcultured. Okay, this subculturing is dependent on the exhaustion of the culture media. When the cells have been utilized all the media, then they will not grow and they will not divide. So at that time, we need to subculture. So now we are going on the secondary cell culture and cell lines. Okay. So what are the secondary culture? Secondary culture is a technique in which the cells of the primary culture are subcultured in a different media. Okay, and these cells are subclone of these primary cell culture cells. Okay, the cells are removed from the primary cell culture and are transferred in the media, in another media, for their regrowth. So that is the known as the secondary cell culture. Okay, when we are going for the secondary culture or subculturing, the primary cells will be converted and they will divide, and due to their division they will create cell lines. Okay, more and more division of these cells of primary cell culture and the secondary cell cultures will lead to generation of cell lines. Okay, so that this might be also the definition of the cell lines. Cell lines are the continuous growth of cells on a medium in the primary plus secondary culture. Okay, that is the cell lines. So this cell lines formation is very important for the growth. These cell lines are requirement of our animal cell culture. That is our basic requirement. So our lecture is over here. In the next video, we'll discuss another lecture. Thank you very much.